Hello everyone, myself Dr. Ghodke Mangesh Sakharam. I am here to discuss about antiamoebic agents. So first we will go through the introduction, classification, different drugs and mode of action of all the examples. So first, introduction. Amoebiasis is the protozoal infection caused due to amoeba, that is Antiamoeba histolytica, which can invade the wall of colon or other parts of body like liver, lungs or skin. The cyst is the transfer stage of the parasite from one person to another, which can occur by the first, direct person to person contact, cyst contaminated clothing, and ingesting cyst which reaches the mouth through contaminated food or drinks. Now we'll discuss the life cycle of amoeba. The cyst passes unchanged through the stomach and proximal portion of intestine to a site where pH is neutral or alkaline. At this stage, cyst increases its activity, rupture its outer envelope, escape as naked protoplast, that is metacyst, which undergoes cytoplasmic division to form metacystic tropozoite. These metacystic tropozoite are the nucleus or nuclei of metacyst. These tiny nucleus are carried by fecal stream to cecum, where they grow into full-size tropozoite. This tropozoite is the active multiplying stage which will form the colonies and attack the tissue and produces lesions. It derives its nourishment from the host tissue and multiplies by binary fission. This tropozoite stage continues to remain in the host tissue, and it is the predominant form seen in the dysentery stool. When the dehydration of feces occurs in the lower portion of the colon, the tropozoite expel all the indigested substance from it and round up to form precyst. This precyst subsequently secretes a thin and strong membrane around itself to become a cyst, and this cyst it will again transfer through the food or drink to the another person. Classification of antiamoebic agents. These are widely divided into three categories. The first, agents effective against extraintestinal form. Examples are natural drugs like ipecac alkaloids, emetine, dihydroemetine. Second, agents effective against intraintestinal form like 4-hydroxyacetonide. Examples are diloxanide furoate and quinfamide. Third, agents having mixed action like nitroimidazole derivatives. For example, metronidazole, tinidazole, ornidazole, furazolidine or furazolidinone. The first agent is the agents having mixed action. The example is metronidazole derivative. So this is the example structure of metronidazole having IPC nomenclature 2-methyl 5-nitroimidazole 1-il ethane 1-ol. The second one is ornidazole. The IPC nomenclature is 1-chloro 3 in bracket 2-methyl 5-nitroimidazole 1-il propane 2-ol. The third example is tinidazole. This is the structure of tinidazole and IPC nomenclature is 1 in bracket 2-ethyl sulfonyl ethyl 2-methyl 5-nitroimidazole. The mode of action of All these nitroimidazole derivatives are discussed as below. All these agents, they are having nitro group at the fifth position of its structure. So they are known as nitroaryl derivative. This nitro group undergoes reduction and subsequent oxidation to form superoxide radical anion. This superoxide radical anion undergoes reduction process to become hydrogen peroxide. Now this hydrogen peroxide undergoes homolytic bond cleavage to form hydroxy free radical. These three SRA, that is superoxide radical anion, hydrogen peroxide and hydroxy free radical, they are collectively known as reactive oxygen species which inhibit the DNA synthesis and prevent the growth of amoeba. The side effects of all these agents are like abdominal cramps, nausea, vomiting, diarrhea, vertigo. The next examples is furazolidine or furazolidinone. The IUPAC nomenclature is 3,5-nitrofuran, 2-ill-methylene amino oxazolidine 2-one. The mode of action is this drug's mode of action is not fully understood but it may be mutagenic or carcinogenic. The metabolic reaction products form of this drug damage the DNA and inhibit protein synthesis. Agents effective against intestinal form like dioxazonate furan. This is the structure of dioxazonate furan and the IUPAC nomenclature is 4-2,2-dichloro N-methyl acetamido phenyl furan 2-carboxylate. The mode of action is it is similar to that of chloramphenicol. It binds to 50S ribosomal subunit and inhibits protein synthesis. It also inhibits polymerization of amino acid to the peptide chain, that is cell wall of parasite. Thank you.